Hey everyone, welcome back to our lecture series covering Tolero's Foundations in Microbiology 12th edition. This time we're covering chapter 5, so let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
Welcome back. Looks like chapter 5, we're covering a survey of eukaryotic cells and microorganisms. So let's go ahead and get started here. A quick history of eukaryotes. Eukaryotic cells, remember, they appeared a little bit later than prokaryotic cells. Eukaryotic cells appeared about 2 billion years ago, and they evolved when larger prokaryotic cells engulfed smaller prokaryotic cells to give rise to that nucleus, right? And now it expands upon that though, because now we know of this endosymbiont theory that the organelles, the mitochondria and the chloroplasts, they came originally from ancient bacteria that were engulfed by early eukaryotic cells. Isn't that neat? I'm going to give you some evidence for this endosymbiont theory in a little bit, but it's fascinating to think that the mitochondria in each and every one of our cells originated as an independent bacterial cell once upon a time. And the chloroplasts that are found inside of photosynthetic organisms were once upon a time cyanobacteria. Remember cyanobacteria? Photosynthetic bacteria that were engulfed by those early eukaryotic cells and then gave rise to the chloroplasts. It's so interesting. So if I ask you on an exam or something, what was the endosymbiont theory about? You would say it's the origin story essentially of the mitochondria and the chloroplasts. It's evidence that shows that the mitochondria and chloroplasts of eukaryotic cells originated as bacteria way back when. So here you can see a little bit about what I'm talking about, this origin story for the mitochondria and the chloroplasts. You can see that there was this early eukaryotic cell that developed a nucleus, and then that cell engulfed aerobic prokaryotes like a bacteria, and then that bacteria gave rise to the mitochondria, and then plants and other photosynthetic algae and such also engulfed cyanobacterium, photosynthetic bacterium, which then gave rise to the chloroplasts. So now we have modern day animal cells on the left and plant cells on the right. And remember, both types have mitochondria, but only the algae and the plant cells have chloroplasts. But either way, we know where those originated. They originated as ancient bacteria that used to live on their own that were engulfed. Isn't that neat? And I'm going to give you a bunch of evidence for this theory. Remember, a theory is not just a guess. This is where scientists truly believe that these came from ancient bacteria. I'm going to give you a bunch of evidence for that in a little bit. This table here is helpful to remind us about the eukaryotic microorganisms that are studied by microbiologists. Remember that some are always unicellular. So, for instance, the protozoa are pretty much always unicellular, though a few are colonial. Do you know what colonial means, by the way? Colonial organisms are technically multicellular, but they don't form distinct tissues. They don't form tissues with different functions. Next, there are some organisms, eukaryotic organisms, that may be unicellular, colonial, or actually multicellular. These include the fungi and the algae. And then there are some eukaryotes that we study as microbiologists that are always multicellular, except for their reproductive stage. These include the helminths, which are the parasitic worms, and arthropods, which are actually usually biting insects. These are animal vectors of disease. Here you can see a nice cartoon of a eukaryotic cell, And look how complex this is compared to the prokaryotic cell we studied in chapter 4. You've got all these different membrane-bound organelles. And by the way, what's the main membrane-bound organelle? Can you beat Wicket? That's right, Wicket. The main membrane-bound organelle is the nucleus right here, and it has a membrane around it. In fact, it's a double membrane called the nuclear envelope. In addition, eukaryotic cells have other membrane-bound organelles that prokaryotic cells do not. So they have the nucleus, they have the rough endoplasmic reticulum, the smooth endoplasmic reticulum, they have the Golgi apparatus, they have the mitochondria, the chloroplasts, they have lysosomes, okay, and all of these are membrane-bound organelles that prokaryotic cells just don't have. So remember though, eukaryotic cells have certain features that all cells have. Remember, some DNA, cytoplasm, a plasma or cell membrane, and these little blue dots that are everywhere that represent ribosomes. And of course, some eukaryotic cells have locomotor appendages, appendages for locomotion, like a flagella, or even the shorter beating cilia. Now, I'm not going to go through all of the structures of a eukaryotic cell and what they all do, because we covered that in Biology 1, right? During Biology 1406 for us. We covered the cell and all the parts of the cell, but I am going to focus on parts of the cell that might be interesting when studying microorganisms. So let's start with the flagella, right? The flagella is a long, sheathed cylinder containing microtubules. And these microtubules are arranged in bundles. This is called the 9+2 arrangement because let me show you something. There are 9 doublet bundles. You see, 1, 2, 3, 4, 5, 6, 7, 8, 9 doublet microtubules surrounding 2 singlet microtubules. And these microtubules, look at this, these microtubules span the length of the flagella or cilia, right? So this is why the eukaryotic flagella and cilia are known to have a 9+2 arrangement of microtubules. And this is covered by membrane. The plasma membrane or the cell membrane actually coats the entire flagella. It's 10 times thicker than a prokaryotic flagella. And do you remember from chapter 4, I told you something important about eukaryotic flagella. Eukaryotic flagella beat back and forth. Remember that? So I want to show you that real quick. Eukaryotic flagella whip back and forth. They like think of a tadpole, right? Or if you've ever seen an image, a microscope image of a sperm cell swimming, right? It just beats back and forth. Remember how that's way different from how prokaryotic flagella work. In a prokaryote, the flagella literally spins, and it's not made up of microtubule proteins. It's made up of the protein flagellin, right? So eukaryotes can either whip their little tails and swim forward, or they could even do this weird twiddle where they can kind of pull themselves in the opposite direction. And that's how locomotion works for flagella. Now, cilia, on the other hand, are much shorter. Cilia have a similar overall structure to flagella because they have that 9+2 arrangement of microtubules inside, but they're shorter and more numerous. They're found only in certain protozoa and certain animal cells. Did you know that you have cilia in your body? One example is in your lungs, the trachea. You have little ciliated cells that line the insides of your lungs that beat, and that beating actually expels debris and dust and spores and other contamination from your lungs. Isn't that neat? So you actually have ciliated cells in your own body. However, here we're mainly going to talk about cilia as they're found on protozoa. Remember those single cell animal-like creatures that are eukaryotes? We're going to talk about them. Okay. And the cilia for protozoa function in motility, feeding, and even filtering. Here you can see the most popular ciliated protozoa of all. This right here is called paramecium. And if you look very closely, you can see the little hairs. It looks like it's completely covered by cilia. And cilia beat, but they don't beat exactly like flagella. I want to show you something. Cilia don't just beat back and forth like a fish tail. Instead, they do this almost like a breaststroke. They do a power stroke where they propel forward, and then they do this recovery, which reminds me of the breaststroke. If you've ever seen a diver or swimmer, I should say, a swimmer do the breaststroke, they put a lot of power into that stroke, and then once they finish that stroke, they just recover for another power stroke, right? So you should know the difference between the motion of the cilia and the flagella. Flagella whip back and forth, cilia do a power stroke. Or imagine if you're rowing a boat, right? You got the power stroke and then recovery, power stroke and then recovery, like that. Now, on the outside of many of these microorganisms that are eukaryotic, you also have the glycocalyx, which is the outermost boundary that comes into direct contact with the environment. Usually, it's made up of polysaccharides, meaning sugars. It can appear as a network of fibers, a slime layer, or even a capsule. What do you use it for? What do microbes use their glycocalyx for? Adherence, protection, and even signal reception. Beneath the glycocalyx, what do these microorganisms have? Fungi and most algae have a thick, rigid cell wall surrounding the cell membrane. Fungi usually have a cell wall of chitin, while algae usually have a cell wall of cellulose, though it can be different substances as well, depending on the species you're talking about. Protozoa, remember protozoa? Um, Protozoa are the single cell animal like eukaryotes. Protozoa and a few algae and all animal cells lack a cell wall and only have the cell membrane. So you and I, our cells lack a cell wall, and so do some protozoa and a few algae. So for instance, let me give you an example. Our paramecium friend here is lacking a cell wall, right? And why do these microorganisms require a cell wall? Well, uh, to provide a rigid structural support to the cell, to help with the shape of the cell. I told you that fungi have this cell wall of chitin and sometimes cellulose. Algae can have a variety of different cell walls. Oftentimes it's cellulose, but it can be pectin, it could be mannan, different mannans, it could be silicon dioxide, like glass, like actually glass. Think of diatoms if you've ever studied the beautiful diatoms in the past, and calcium carbonate. All of these are types of cell walls found in different species of algae. And of course, like all cells, these eukaryotic microorganisms have plasma membranes, right? It's a typical barrier of phospholipids. Remember the phospholipid bilayer with proteins embedded in it. Sterols can confer stability. It serves as a selectively permeable barrier in transport. It can also function in surface adhesion, secretion, and signal transduction. So that leads us to our first concept check. Which part of the eukaryotic cell is in direct contact with the outside environment? Is it the flagella, the cell wall, the glycocalyx, or the cell membrane? This question is really, I don't know if I don't know if I, mm, I don't know if I really like this question, because if you think about it, a flagella is in contact with the outer environment. A cell wall could be, especially if you don't have a glycocalyx, and the cell membrane could be, especially if you don't have either of the above. but I'm guessing what what they want is answer choice C. So I really don't like this question, and it probably won't appear on any of my exams, but I guess they're talking about the outermost layer, which would be the glycocalyx. And here are some organelles as well as their functions that are found in eukaryotic cells. Again, we're not going to cover every one of these, the nucleolus, the Golgi apparatus, lysosomes, vacuoles, all that. We should have covered that in biology 1. So I'm going to leave that for now. And like I said, I'm just going to focus on what's unique about microbiology, what's different about these organisms, and where we can expand on what we've learned from biology 1406, biology 1. By the way, if you're curious and you want to learn this, if you want to know what is the Golgi apparatus, what is the endoplasmic reticulum, why don't you go ahead and watch my video here right above my head. I'm going to put a card, and you can watch my video on from my biology 1 course for science majors, where I go into great depth about how all these different organelles work, right? So you can catch up if you want to, but if you're already taken bio 1, then we can carry on with what's unique and different about these organisms. And remember, eukaryotic organisms, the cell divides by a process known as mitosis, okay? Mitosis is how cells divide. And this is different than prokaryotes. Do you remember that I mentioned in a previous chapter that prokaryotes, they don't divide by mitosis. They divide by a process known as binary fission. And I'm going to explain binary fission in another chapter. But if you're not familiar with mitosis and you just want to learn about the phases of mitosis and how mitosis works, I've got another short little video right here where you can click on, and that explains mitosis for you nicely. But I'm not going to recover it here in this class. But just be aware that eukaryotic cells divide by mitosis, while prokaryotic cells divide by binary fission. Now, one thing I will touch on are the lysosomes. Do you guys remember lysosomes from biology 1? If not, these are eukaryotic organelles. They are essentially vesicles full of enzymes. And these enzymes break down organic molecules. So they're called hydrolytic enzymes. They originate from the Golgi, all right? And what they're involved in, what they do is they can digest intracellular food particles. Okay, they can digest food. So think of the lysosome as the digestive organelle of a cell, of a eukaryotic cell. And so what happens is a lot of these single cell eukaryotes, like protozoa, for instance, they will bring food in through phagocytosis. Do you guys remember what that is? Let me show you something. Okay, let me show you. Imagine if this is a eukaryotic single cell creature, like an amoeba. The amoeba will find some food, and the plasma membrane will invaginate like this, right? The plasma membrane will bring the food in. And then the plasma membrane wraps around the food. By the way, this is the process known as phagocytosis, which is a type of endocytosis. And then the plasma membrane wraps around the food, and then pop, the food comes inside of a membrane. That membrane now is called a vacuole. So this is known as a food vacuole because it's food inside of a plasma membrane. Now, don't we want to digest this food? So what happens next is our lysosome friend, right? The lysosome, which is a vacuole full of digestive enzymes, will fuse with the phagosome, which is this vesicle full of food particles, and then you'll have digestion, right? Because now you've introduced the enzymes from the lysosome to the food particles, and you have digestion. And now you've digested those organic molecules, and you can use it to grow, right? That's the nutrients you need to build your own molecules, your own biomolecules. Isn't that neat? So that's something we should be aware of, that these little organisms are doing, utilizing lysosomes to digest food that was ingested by phagocytosis. And do you remember that I had mentioned that phagocytosis is how the mitochondria and the chloroplasts came to be? It's really their origin story. So let me show you the structure of a mitochondria as well as a chloroplast. And then I'm going to tell you a little bit about what evidence we have towards the endosymbiont theory. It's really neat if you think about it. So first of all, do you remember the mitochondria, the cheesy saying that it's the powerhouse of the cell? Well, this is because it functions in energy production to produce ATP, right? It produces ATP. And do you remember the structure of a mitochondria? On the left, we have an actual image of a mitochondria with an electron microscope. And notice how in the inside it has this, convoluted, twisty, turny membrane. So let's look at the cartoon here to understand how it works. Remember, the mitochondria has a smooth outer membrane, a convoluted inner membrane. And remember, the folds of this inner membrane are called cristae. Then the fluid in the inside of the mitochondria is called the matrix, right? That's the fluid in the center of the mitochondria. Now, why do we say that we have a lot of evidence that mitochondria originated as a bacteria once upon a time? Well, take a look here. First of all, look at this. Mitochondria possess their own DNA. And not only is it DNA, it is circularized DNA. Let's see if you can beat Wicket. What else has circularized DNA that we've studied? That's right, Wicket. Bacterial cells, right? Prokaryotic cells have circularized DNA. Remember, it's double-strand DNA with no beginning and no end. Well, the mitochondria has its own DNA. And yes, that's an example of extra chromosomal DNA. DNA that is not part of your actual 46 chromosomes, right? Isn't that neat if you think about it? Did you know that, your 46 chromosomes are not the only chromosomes in the cell? That there's DNA within each and every one of your mitochondria? Isn't that neat? Anyway, this DNA houses genes, and did you know that they have sequenced these genes and done sequence analysis, and these genes match up best with prokaryotic genes, not anything human, not anything eukaryotic. So that's another piece of evidence. And what are these little blue dots everywhere? Take a look. What are these blue dots? Well, it looks like they are ribosomes. And by the way, what type of ribosomes are these? 70S ribosomes. Now, do you see something strange? You and I are eukaryotes, right? Eukaryotes have what type of ribosomes? Have you been paying attention? That's right. Eukaryotes possess 80S ribosomes. But what type of ribosomes are found in the mitochondria? That's right, 70S ribosomes. Who else has 70S ribosomes? Can you beat Wicket? That's right, Wicket. Bacteria, right? Bacteria possess 70S ribosomes. And guess what? The mitochondria has its own ribosomes, and those are the same ribosomes as bacterial ribosomes, whereas the rest of your cell has the 80S eukaryotic ribosomes. Isn't that weird? And by the way, you know the mechanism by which mitochondria produce ATP? It's the same way, it's the same mechanism by which bacteria produce ATP, using ATP synthase, using the electron transport chain. So isn't that interesting? Okay. And by the way, do you know where mitochondria come from? How do your cells produce mitochondria? Because each and every one of your cells has numerous mitochondria inside, right? So, where did those mitochondria come from, do you think? What part of your cell makes mitochondria? I'll let you think, right? Actually, that's a trick question because mitochondria come from mitochondria. You know what mitochondria, if this mitochondria divide. So this little mitochondria, if it wants to divide, it'll copy its DNA, separate the two DNA molecules, and then it will undergo basically binary fission. a very similar asexual form of division to bacteria. Isn't that neat? So these little guys divide independently in the cell. Isn't that neat? So I hope I've convinced you. I mean, everything from having one circularized chromosome to the genes on that chromosome matching up with prokaryotic genes to 70S ribosomes to the mechanism of ATP action and the mechanism by which these things divide is all mirroring bacteria. So there's ample evidence that these mitochondria originated as bacteria. Now, through a process known as genome reduction, they have lost many, many genes over the millennia. So they can no longer be bacteria. Like if you were to pluck one of these mitochondria from a cell and put it in a dish and expect it to grow, it can't grow on its own anymore. It's lost its ability to grow independently because of genome reduction. It's lost various genes it doesn't need over years and years and years. So essentially, this is a remnant of a prokaryotic, a prokaryote from the past. Isn't that neat? That each of us then is a hybrid organism, if you think about it. You and I are all hybrids, like kind of like a Toyota Prius or something. A Prius is a hybrid between a gas engine and an electric engine, and you and I are kind of a hybrid between eukaryote and prokaryote. Isn't that neat to really think about it? Each one of our cells has little ancient prokaryotic zombie cells inside. It's really fascinating, actually, to think about. And the chloroplast is no different. Remember the chloroplasts? Chloroplasts have these stacks of granum where photosynthesis happens. But remember, look at this. They also have a single circularized chromosome with genes that sequence nicely with prokaryotic genes. They have 70S ribosomes as well. The way they do photosynthesis is similar to how cyanobacteria undergo photosynthesis. And how do you get more chloroplasts? That's right, these guys divide on their own. So the origin of the chloroplast is also prokaryotic, except this time it was a cyanobacteria, a photosynthetic bacteria that was phagocytosed by that early eukaryotic cell billions of years ago. Isn't that interesting? So hopefully you understand the endosymbiont theory and how it is evidence towards the prokaryotic past of the mitochondria and the chloroplasts. Very fascinating to think about. And again, do you remember that I mentioned that ribosomes are made up of protein, but also RNA? The RNA portion of a protein is called ribosomal RNA, or rRNA for short. And the rest of the ribosome is made up of protein. Do you remember that there's a large subunit and a small subunit? Well, the eukaryotic ribosomes are larger than the prokaryotic ribosomes. Eukaryotic ribosomes are called 80S ribosomes. Remember that prokaryotic ribosomes are smaller. They are called 70S ribosomes. And do you remember the function of ribosomes? They function in protein synthesis. All right, so at this point in the chapter, we're going to do a survey of eukaryotic microorganisms. We're going to look at examples of fungi, algae, protozoa, and worms as well. So let's do that next. Let's start with the kingdom fungi. A majority of these are unicellular or colonial, but some have cellular specialization, so they are multicellular. There are about 100,000 species divided into two groups. The macroscopic fungi that you and I know and love, right? Mushrooms, puffball, gills, fungi, shelf fungi, rusts, things that you can actually see. And then the microscopic fungi. These are the ones that are most studied by microbiologists. They have two morphologies. These fungi either have filamentous cells, so they form hyphal cell shapes, long filamentous cells, and these are known as the molds. Molds have hyphal cell shapes. And then there are the yeasts. The yeasts are round, ovid shaped. They're semicircular, and they can undergo asexual reproduction, though yeasts can also undergo sexual reproduction as well. Here you can see the classic telltale sign of mold, right? Everyone's seen mold on fruit. It looks fuzzy, velvety. Well, that's called the mycelia or the mycelium. It's a network of hyphae that form this fuzzy-like surface. Okay, long thread-like cells that make up the bodies of filamentous fungi or molds form a mycelium. So when you see this fuzzy colony, when you see this fuzzy substance on top of fruit or on top of food, you're usually correct to guess that it's a type of fungus. And not just a type of fungus, but a type of mold. And if we look closely at these mold hyphae, you can tell that they are long filamentous cells, and sometimes they have segments called septum, which separate the different cells. You can have septated molds, and then there are non-septated molds as well. Yeasts, on the other hand, remember, are semicircular or ovid cell-shaped cells. They can undergo asexual reproduction, but they can also undergo sexual reproduction as well. Yeasts have a cell wall and lack locomotor organelles, so they don't have flagella or cilia or anything of the sort. And the way that yeast cells divide asexually is kind of neat. They form what's called a bud, right? A small segment swells and grows out from the surface containing a copy of the genome, and that pinches off to form a smaller organism that then grows in size to become a full-size yeast cell. These are also known as budding yeast because they divide by a process known as budding. It grows swellings on its surface called buds, and these buds contain a copy of the genome, which then become separate cells. So you can see this process here. Here's a yeast cell in the process of cell division, and instead of proper mitosis, it forms a tiny bud. That bud pinches off and leaves behind a scar, and that's known as a bud scar. So when you look at pictures of yeast under the microscope, like this is an electron micrograph of yeast, you can not only see the budding here, but you can also see the left behind scars from budding past. Isn't that neat? Now, how do fungi eat? Fungi are never photosynthetic, okay? Fungi are always what are known as heterotrophs. They feed off of organic molecules. They feed off of organic molecules. So a majority of them are harmless saprobes. Saprobes refers to organisms that feed off of dead organic material. So imagine a tree that's fallen over or an animal carcass, right? It's a decomposer. You should think of most fungi as saprobes, as decomposers of dead organic material. However, some of these fungi can live as parasites on living tissue. If you've ever had a fungal infection, right? If you ever had athlete's foot or ringworm, you know what I'm talking about. That's a type of parasitic fungal infection. And by the way, fungal infections are called mycoses. So mycoses are fungal infections. Okay. So those are the two main nutritional types for fungi: saprobes, the decomposers, and the parasites living off of living tissue. Here you can see a very common mold. This here is a close-up of the mold under a microscope on the right. This is Penicillium notatum. And I bet you've seen this before on citrus fruits. It loves to grow on citrus fruits, and it forms this cyan green color mold, mycelium on the surface of citrus fruits. So this would be an example of saprobic nutrition, right? Breaking down dead organic material. However, in this slide, you can see Trichophyton rubrum, which is a parasitic type of mold. Here's a close-up of the mold here on the right, as well as its little spores. This mold causes athlete's foot, right? So you can see that it's a parasitic type of mold. So again, when we're talking about molds, we're talking about hyphal cell shapes that come together to form a mycelium. The mycelium is that fuzzy surface that you see. It's a woven intertwining mass of hyphae. Remember, some of these hyphae can be septated. They have cross walls or septa. Yeasts, on the other hand, are not filamentous. Yeasts are semicircular or ovid in shape. So again, on the left, you can see molds. This is a septated mold on the far left and a non-septated mold on near the center here. And on the right, you can see how that's really different uh, with cell shape to yeasts. Yeasts are ovid shape and they divide by budding. Molds are filamentous shape and they divide by forming spores, asexual spores, though they can also reproduce sexually as well. So I'm going to explain that in a little bit, how molds can divide asexually as well as sexually. And we already discussed the asexual reproduction of yeasts via bud formation. We're not going to talk about the sexual reproduction of yeasts. All right, let me demonstrate for you how these molds can reproduce asexually. This is the asexual mechanism of reproduction for this mold. And I believe this mold is called Rhizopus, which is the common bread mold. So what happens is a spore is disseminated, and this is an asexual spore called a sporangiospore, is disseminated from the fungus. We'll get back to what these structures are on the fungus in a minute. But these asexual spores disseminate, and you should write down that disseminate means scatter, usually through the air. The spores then land on a substrate. So let's say, I don't know, bread, for instance, something yummy like bread or fruit. The spore will then germinate, forming a germ tube. This means that the mold is growing out from the spore. It forms those filamentous cells called hyphae. This is known as the vegetative hyphae, which feeds on the substrate. At one point, surface hyphae form and grow tall up into the sky. These are surface hyphae that grow up into the sky. And at the tips of these aerial hyphae, they're called, or surface hyphae, aerial hyphae, little caps form. This is called a sporangium. It's a cap in which asexual spores called sporangiospores form. And at some point, the cap ruptures, allowing the sporangiospores to disseminate again. Isn't that neat? So this is known as the asexual life cycle of Rhizopus stolonifer, the common bread mold. And this is typical of how molds asexually reproduce. And again, I want you to notice something. When the spores, the asexual spores, are inside of this little cap structure for molds, this is known as a sporangium. See, sporangium. And the sporangiospores are inside. However, there are also molds that don't form a cap like that. These are called conidia, and these conidia form conidiospores. These are free spores not enclosed by a spore-bearing sac. Do you remember back here? Look at this. Do you remember our penicillium friend over here? This is an example of a conidia with conidiospores. See how the little asexual spores are not within a cap? Well, penicillium does not form a cap, right? So this is known as a conidia. with conidiospores, whereas Rhizopus forms a cap. So that's a sporangium containing sporangiospores. That's one main difference between the asexual spores of different species of fungi, of molds. Okay, so again, just to summarize, take a look at this. On the left, you have the sporangium. On the right, you have conidia. Notice how on the left, the sporangium forms this cap, and within it are those spores, those asexual sporangiospores. And yes, this is an asexual structure. And on the right, we have the conidia, which is also an asexual structure. However, look, these conidiospores or phyalospores are asexual and they are not within a cap. Does that make sense? So, why do I stress this? Well, because when you're identifying mold under a microscope, some molds have caps, some molds don't. And by looking at the mold under the microscope, you can try to guess the identity of that particular species. Isn't that neat? So, for example, when I see little conidiospores that look like this, I automatically guess it's penicillium because that's what the spores of penicillium look like. So now that we've talked about the asexual spores of molds, let's talk about the sexual spores of different fungi. These are spores that are formed following fusion of two different strains and formation of a sexual structure. The sexual structure can be a zygospore, an ascospore, or a basidiospore, depending on the species we're talking about, depending on which group of fungi, right? Different fungi are classified based on the sexual spores they produce. So right here you can see sexual spores and spore-forming structures are one basis for classifying fungi. So for instance, there's an entire group of fungi called the zygospores, and that's because they form the sexual structure a zygospore. In fact, do you remember our Rhizopus friend who forms the sporangium with the sporangiospores inside? This is the sporangium of Rhizopus, the bread mold, okay? And remember, that cap breaks and releases those sporangiospores. Those sporangiospores land on bread, they germinate, okay? And by the way, what happens is two different hyphae, a positive mating type and a negative mating type, touch. And when those two different creatures touch, when those two different hyphae touch, they fuse. Contact of mating hyphae. See, they contact, they fuse, forming a gametangia. This leads to fertilization, right? So it's a sexual type of reproduction to form the zygospore. This is a sexual structure, right? So when you see this under the microscope, this is one hyphae with the one mating type, like the positive mating type. This would be the negative mating type hyphae of the other organism. And what's in the middle? This is known as the zygospore, the sexual spore. This zygospore then matures, germinates, and produces its own sporangium. Isn't that neat? So this is the sexual life cycle of the Rhizopus, whereas this over here was the asexual life cycle of the Rhizopus. Same creature, just different mating type. Okay, there's also what are known as ascospores. Again, a female mating type, ascospore hyphae, fuses with a male mating type, ascospore, to form this ascogenous hyphae. And at the tip of the hyphae, you form asci, which are essentially sacs. Within the sacs, meiosis occurs, and when the sacs rupture, opposite mating types are released, called ascospores. So when you think of ascospores, think of what are known as sac fungi, which form spores within a sac, sexual spores within a sac. And then there are the mushrooms and organisms like that. These are called the basidiospores. Again, this is how they mate. You have the negative mating type hyphae, the positive mating type hyphae. They come together and fuse to form to fertilize. Right, during fertilization, the positive mating type and negative mating type fuse. This forms a mycelium. A young mushroom button forms to form the fruiting body of the mushroom. Then underneath the cap of the mushroom are these little gills. And if you take a close look at the gills, they have these structures called basidia. And at the tips of these club-shaped basidia form those basidiospores. And that's how the mushrooms actually undergo sexual reproduction. Isn't that neat? So again, remember what I said, that the kingdom fungi, the eukaryotic fungi, are subdivided into several phyla based on their what? Their type of sexual reproduction. There is a phylum Zygomycota that forms those zygospores that I showed you. There is Ascomycota that forms ascospores. There's Basidiomycota that forms basidiospores. There are even Chytridiomycota that form flagellated spores. And then there are these weird ones, this fungi that reproduce only asexually. And again, how do we identify fungi in the lab? Well, first we isolate a sample. We grow it on specific media. And what are we looking for? We're looking for the asexual spore forming structures. Remember, like the sporangium or the conidia? We're looking at hyphal type. Is it a septated hyphae or a non-septate hyphae? Colony texture. So when it grows on a plate, what's the texture look like? Is it pigmented? Like for instance, is the mold green or is the mold gray or black? Physiological characteristics, so what is it able to break down or do, and even its genetic makeup. So let's delve a little deeper into these fungal groups and characterize them a little bit. Phylum 1 of fungi, remember, are the Zygospores, also known as the zygomycetes. Remember that the sexual spores are called those zygospores, whereas the asexual spores are usually called sporangiospores and sometimes conidia. Most of these guys are free-living saprobes, though some can be animal parasites as well. Remember that the example I showed you was Rhizopus, which is the common bread mold. So let's take a look at that. Here's Rhizopus up close. And you can see those sporangium full of sporangiospores here. This is the asexual reproductive structure. Next, the second phylum of fungi, the Ascomycota, also known as the sac fungi. Most produce ascospores in an asci, so they form these spores within a sac. Next, we have the third phylum of fungi, the Basidiomycota. These include familiar macroscopic forms such as mushrooms, puffballs, toadstools, things that you've seen before probably. But it also includes some microscopic plant pathogens called rusts and smuts that attack and destroy major crops. So they have agricultural implications. Remember, we covered that sexual reproduction occurs by formation of a basidia and those basidiospores on the gills of the mushrooms. And the common mushroom is an example. You've seen mushrooms before. Now you can look at the underside of those gills and take a look at those sexual spores and understand just how they were formed. That leads us to group 4, the Chytridiomycota. These are tiny primitive fungi called chytrids, commonly called chytrids. They can either be, some of them are actually single cell fungi, though they can also form clusters and even colonies, though they are usually characterized by special flagellated spores called zoospores and gametes. Most of these chytrids are saprobes, free living in soil, water, and decaying matter. Here you can see some chytrid cells attacking a diatom and type of algae. And again, we're going to be studying fungi because of their importance in a number of different fields, right? Some of them cause human diseases called mycoses. They can cause allergies. They can produce toxins, right? They can also, on the other hand, attack crops. So they have agricultural importance and they can cause food shortages by destroying those crops. However, they are also beneficial. They are sources of antibiotics, alcohol, organic acids, vitamins. They decompose, which is so important to decompose in the ecosystem. And they're also used in fermenting foods, right? And in genetic studies. In fact, many foods are produced by fungi, like beer and wine are produced by fungi, and several forms of bread, right? So they have many beneficial impacts, adverse impacts. So it's very, very much worth studying fungi this semester. So that leads us to concept check number 5, and that'll lead us to our break time with Gizmo and Wicket. So let's go ahead and answer this question. I think we know the answer to this question, right? Fungi are generally classified according to what? Their type of sexual spore. Isn't that correct? The zygospores, the basidiospores, remember all that? So it's the type of sexual spore. That's fantastic. Answer is A. So with that, let's take a quick break time with Gizmo and Wicket. Come back strong, finish off this chapter. What do you guys think? All right, welcome back from break time with Gizmo and Wicket. Let's carry on with our discussion of the protists. Remember, protists are eukaryotes, usually single cell eukaryotes, and they include the algae, which are photosynthetic, and the protozoa, which are never photosynthetic. The protozoa are called animal like because they are heterotrophs, which means they feed on organic material. Algae do photosynthesis. The algae are eukaryotic organisms, usually unicellular, though some can be colonial, kind of like seaweed and kelp, right? That's an example of colonial algae that photosynthesize with chlorophyll A. Okay, protozoa, on the other hand, are never photosynthetic. They are unicellular eukaryotes that lack tissues and share similarities in cell structure, nutrition, life cycle, and biochemistry. So let's discuss algae and then protozoa. Starting with the algae, again, algae are photosynthetic organisms. In fact, they produce much of the oxygen on the planet through their photosynthesis. And again, these algae have chlorophyll A, but they can also have other pigment molecules as well. And many have a cell wall of some sort. Here's an example of a flagellated algae on the left. Here's a depiction of a flagellated algal cell. Notice that algae have chloroplasts inside. On the right, you have another form of algae. This is known as a diatom. Diatoms have different beautiful shapes and sizes. This one happens to be circular, but there are rectangular ones, square ones, triangular ones. I've even seen some that look like stars. They've got beautiful glass shells. And this is another form of algae. Many algae are free living in fresh and marine water. This makes up plankton, right? Have you ever heard of plankton? And I'm not talking about SpongeBob, right? Plankton makes up the bottom of the food chain. All of the other aquatic life uh, requires plankton, right? Depends on plankton for feeding, right? Because plankton makes up the bottom of the food chain. These are producers that photosynthesize. All other aquatic life feed off of that too in order to grow. It's the basis of the food web in the aquatic habitat. Not only that, but these algae, they're heavy lifters when it comes to producing oxygen for us to breathe. through their photosynthesis that they conduct. Here you can see a beautiful type of algae. This is a two mirror image semi-cells connected by a narrow isthmus. This organism is known as Microasterias truncata. This is a type of algae. There are so many types of algae. It would be impossible to go through all of them in one course. And on the right, you can see an algal bloom of what are known as dinoflagellates in Puget Sound. All right, moving on to the protozoa. Remember, protozoa are never photosynthetic. It's a diverse group of 65,000 species. They vary in shape. Most are unicellular. Colonies are really rare. Most are harmless, free living in moist habitats, but some are animal parasites, and these are the ones that are most interesting to us microbiologists. Many of these parasites can be spread by insect vectors. When you see this term vector, you should think a living transmitter of disease. So the classic example of a vector is a mosquito. Mosquitoes are just, they're the ultimate vector. They bite one person, pick up a bug, and then bite another person, and now the other person's caught the bug, right? And they caught the germ. So vectors are living transmitters of disease. Any biting insect is usually some kind of vector for some disease, right? Like think of ticks. You can get diseases from ticks. You can get diseases from mites. You can get diseases from lice, right? All of these are vectors, insect vectors. Okay, all of these are heterotrophs. Remember, they feed on organic material. They are not photosynthetic, so they do not have chloroplasts. And by the way you classify protozoa is based on their locomotor structure. Some have no motility at all. Some have flagella. Some have cilia, and some have pseudopods. Um, we covered flagella and cilia, but we haven't covered pseudopods. Pseudopods are extensions of the cytoplasmic membrane, right? Think of an amoeba. Remember, an amoeba is like looks like a blob, right? And the way this blob moves is by extending its cytoplasm. That's called pseudopod motility. Okay, so depending on the type of motility, that's how protozoa are classified. And many of these protozoa, they have two different states of being. They can either exist as trophozoites, which are the motile feeding stage. This is the active stage of the protozoa, and they can also form a cyst stage. This is a dormant sleeping stage, right? When unfavorable conditions are around for growth and feeding, they form a cyst. So they kind of lay low, right? So many protozoa are known for forming cysts when the going gets tough and forming trophozoites when the going gets nice, right? All of these can reproduce asexually, either by mitosis or multiple fission, but they can also reproduce sexually by conjugation. In fact, here you can see the cyst formation of a typical protozoa. Remember that the active motile feeding stage is called the trophozoite stage. When the conditions get poor, so what do I mean by that? Let's say remember they like to live in moist environments. Let's say the environment's drying or there's a lack of nutrients. Well, those are unfavorable conditions, right? In this case, the cells round up, lose motility, and an early cyst wall forms. Then this organism becomes a mature cyst, which is a dormant or resting stage. And then when moisture and nutrients are restored, so we're talking about the good times come back, the cyst wall breaks open and the trophozoite is reactivated to feed and grow once more. So let me give you a couple examples of important protozoan pathogens, starting with these flagellated ones called trypanosomes. These trypanosomes are known for their long flagella. The genus Trypanosoma contains Two very important pathogens, Trypanosoma brucei, which causes African sleeping sickness, and Trypanosoma cruzi, which causes Chagas disease. Again, these trypanosomes possess long crescent-shaped cells with a single flagellum. They are blood diseases, so they occur in the blood during infection. And they are transmitted by those blood-sucking vectors, these insects that bite. Trypanosoma brucei is spread by the tsetse fly. Trypanosoma cruzi is spread by the kissing beetle. In fact, let's take a closer look at Trypanosoma cruzi and how it causes Chagas disease. There is a beetle, and this is called the kissing beetle. Its proper name is the reduviid bug, or kissing beetle, I'll call it. The kissing beetle picks up the trypanosome. This is, again, the blood-borne infection. It picks up the trypanosome either from domesticated pets, such as this cat, or it can even pick it up from armadillos or possums or other wild animals. They have to be usually mammals. They pick up the trypanosome, and they're known as kissing beetles because they like to bite near the mouth, right? Because they can sense where the mouth is, they sense CO2, they bite you close to the mouth. When they do, they essentially secrete excrement, they poop, and then when you wake, you have an itch, you scratch that itch, you introduce the trypanosomes into the cut, into the wound, and that gets into the body, and that's how infection occurs. And that's a devastating infection because at first all that happens is you get like a flu-like symptoms, but then they can resolve and the trypanosome can go dormant for a while. And then years or even decades later, it can manifest in some horrible conditions, uh, including heart attack and death, right? So this is a disease that's most common in Central America, okay? And there have been cases of it in the lower states, and so we need to screen for trypanosomes in the blood, especially if you are a blood donor or if you are an organ donor. We need to screen for these types of blood parasites because they can have devastating outcomes for the patients. Another important protozoan pathogen is a type of amoeba. And remember, amoeba move around with pseudopods, these fake feet, right? They're extensions of the cytoplasm. A great example here is entamoeba. Entamoeba histolytica causes a condition known as amoebic dysentery. Ingested cysts germinate in the small intestine. The trophozoites, remember the active state, migrates to the large intestine to grow. They can then invade the liver, the lungs, the skin, and common symptoms include gastrointestinal disturbances. So here's how it works. You ingest the cysts, remember the dormant stage of this amoeba. You ingest it through the food and water. It goes down into the intestines. The cysts open up to release the trophozoites. The trophozoite matures and then later on forms into cysts and releases through the feces. If these feces were to contaminate food or water, let's say from sewage contamination, and are re-eaten, that's how the life cycle continues, right? So the cysts are secreted in feces, and the cysts, they enter new hosts by contaminated food and water. And again, these trophozoites can get into different organs, but they can cause dysentery. Dysentery means bloody diarrhea, right? This is a terrible form of painful gastrointestinal disease. So that leads us to our concept check. Which of the following descriptions is true of both algae and protozoa? Are both photosynthetic? No. Are both always unicellular? No. Do they both contain mitochondria? Yes, algae, even though algae are photosynthetic, so they have chloroplasts, they also have mitochondria. So this is the correct answer. It's not D because they're not both heterotrophs. Remember, algae are photosynthesizers. So the answer should be C. Yeah, algae as well as plants. Did you know that plants and algae, not only are they known for having chloroplasts and being photosynthetic, but they also have these mitochondria, right? They have mitochondria as well. All right, and lastly, with our survey of eukaryotes, let's look at the helminths, the worms. These are multicellular animals. They have organs for reproduction, digestion, movement, protection. Many of these are parasites on host tissues. They can be human parasites or animal parasites. They have mouth parts for attachment or to digest host tissues. Most have well-developed sex organs that produce eggs and sperm. Now let me tell you about the major groups of parasitic helminths. There are two major groups here: the flatworms, known as platyhelminthes, and the roundworms, known as nematodes. Let's start with the flatworms first. Flatworms, as their name suggests, are flat. They have no definite body cavity. They have a simple excretory and nervous system. And there are two classes of flatworms: the cestodes, which are known as tapeworms, and the trematodes, which are known as the flukes. The tapeworms, we're going to talk about tapeworms in a second. The tapeworms have segmented bodies, while the flukes have non-segmented bodies. The roundworms, on the other hand, the roundworms known as nematodes, as their name suggests, they are round. They're cylindrical and have complete digestive systems. Here we're taking a closer look at the flatworms, the platyhelminthes. On the left, we're looking at the trematode, the tapeworm, and on the right, we're taking a look at the cestode, which is the type of fluke. So on the left, we're looking at the pork tapeworm. And remember when I said it has a segmented body style? See these segments right here? It's a flattened worm, but it has these body segments. And the body segments are called proglotid. It also has a head. The head looks like this, and it's called a scolex. The scolex contains two suckers as well as hooks. The hooks and suckers assist in attachment to the inside of your intestines. Isn't that interesting? So we need to know that the head is called the scolex and the body segments are proglottids. The fluke, on the other hand, here's an example of a fluke on the right. This one's the liver fluke, fasciola. And notice how it has a complete digestive system. It has a mouth all the way over to the excretory pore on the other side. Now, there are about 50 species of worms that parasitize humans. They're distributed worldwide. And oftentimes, the way you pick up worm diseases, and this is why we study worm diseases with microbiology, is that it's those microscopic larvae or eggs that we ingest without even knowing it in the food, soil, water, and sometimes even carried by insect vectors through insect bites. And these afflict billions of humans. Here's an example of a roundworm infection. This is a nematode called the pinworm. You may have heard of pinworm. It's common among children. The pinworm is introduced, the fertile egg is introduced into the mouth, usually by contaminated hands. Once the worm makes its way into the mouth of the human, it makes it down into the GI tract, where it then becomes the adult worm. This is the female adult worm on the left and the male adult worm on the right. And these produce more fertile eggs, which are then excreted in the feces. If you don't wash your hands, let's say you get some feces after using the bathroom, you don't wash your hands and you touch your mouth again, you can reinfect yourself. This is called self-infection. And you can also cross-infect. You could not wash your hands properly after using bathroom, and then interact with other people and transfer those fertile eggs to other children or other people, and then they can auto-inoculate themselves. They can self-infect themselves. So again, this is common among children because children don't have the best hand washing hygiene methods, right, going on, and habits going on. So, that's why it's so common among children. So, this leads us to our last concept check. Are you ready? Parasitic helminths reproduce by cysts, spores, eggs, and sperm. It has to be eggs and sperm because these are true multicellular animals. So, eggs and sperm it is. And that leads us to the end of the chapter. Another longish chapter, but hopefully very interesting and fun. Again, don't get bogged down in all the details. It's nice to know all the details, but I will try to pare it down in the exam review video for you to come. So let's go ahead and hop to the next chapter.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D,